Hello children, this is class 8 biology, chapter 2 reproduction in plants, part 1, presented by Mrs. Sonam Singh. What is reproduction? It is a process of producing young ones from their parents. It is of two types, asexual reproduction and sexual reproduction. In asexual reproduction, a single parent is involved and simple division of a plant body occurs into two or more parts, or it just includes the formation of spores. In sexual reproduction, the two parents are involved and the fusion of male and female gametes also occurs to produce a zygote, which further develops into a seed. Vegetative reproduction can also be considered as an asexual reproduction in which any cell, tissue, or part of an organ of a plant can be used for developing into a new organism. Now, let us study more about the asexual reproduction. It is a process by which a single organism produces genetically identical offspring. This type of reproduction is most common in single-cell organisms like bacteria, but can be in more complex organisms as well. The first in the list of asexual reproduction is binary fission. Binary means two and fission means division. In this, first, the nucleus elongates and divides into two, followed by the division of a cytoplasm across the middle and formation of two daughter cells. Each daughter cell grows as a separate individual. It is the most primitive type of asexual reproduction found in bacteria, fungi, and algae. The next in the list is budding. In the process of budding, an outgrowth called a bud arises from the surface of a parent body. This bud grows bigger with the accumulation of a cytoplasm, and the nucleus also divides, which further passes on to each end. The bud ultimately gets cut off from the parent body and leads an individual life. The budding may be repeated, resulting in one or more chains. Example, yeast. Next comes the fragmentation. In this type of asexual reproduction, the filamentous body of lower plants like spirogyra breaks up into two or more fragments. Each fragment grows into a new individual. Now, let us study something about the vegetative propagation. Certain plants reproduce asexually by their vegetative parts like stem, roots, and leaves rather than by seeds. Such method of reproduction is called vegetative propagation. The vegetative propagation takes place in plants which do not produce seeds or whose seeds cannot germinate. It is very fast, easy, and usually very successful method. It can occur by natural methods like in runners, tubers, rhizomes, and bulbs, even artificially by cutting, grafting, etc. Now, let us study about the natural vegetative propagation by underground modified stems. The picture given here is of ginger, which is a rhizome. Rhizomes are branched, underground, horizontally growing stems. They have distinct nodes and internodes which bear axillary buds. These axillary buds can grow into new plants. Example, turmeric and banana. The picture given here is of potato, which is a tuber. A tuber is a modified, fleshy, and swollen underground stem, which contains a food for the plant. It has many buds called eyes. Each eye grows into a new plant, example, potato tubers. Now, let us study about the vegetative propagation by leaves. Leaves of some plants like bryophyllum and begonia produces adventitious buds on their margins. When the leaf falls on the moist soil, these buds develop into small plantlets. On separation from the leaf, these plantlets grow as independent plants. The fleshy leaves of bryophyllum provide food and water to the developing plantlets. Bulbs are also used for vegetative propagation. They act as food storage for the plant to grow from the bud. Now, the vegetative propagation occurring by the roots. The adventitious roots also help in vegetative reproduction. They become fleshy and tuberous. They have adventitious buds which can be detached and grown into a new plant. Example, sweet potato, asparagus, dahlia, etc. Now, let us compare the advantages and disadvantages of natural vegetative propagation. Advantages includes no need of external agencies like insects, wind, etc. Food is usually present in the vegetative structures. A rapid development of buds into daughter plants can also take place. The new plants produced resemble the parent plant in every way. It involves only one parent. The disadvantages are lack of dispersal mechanism may lead to overcrowding. New plants are less varied. New plants may be less adaptable to changes in the environmental conditions. Now, let us learn about the artificial vegetative propagation. Some new plants are developed vegetatively by the human beings to improve the variety of plants. Such vegetative propagation is called artificial vegetative propagation, done by cutting, grafting, and layering. What happens in cutting? A part of stem is cut and the cut end grows into a new plant when grown in the moist soil. Example, mango, guava, lychee, lemon, etc. In layering, the stem of a plant is bent down until it touches the soil. The stem is then cut once it develops roots and grows into a new plant. Example, lemon, rose, and jasmine. In grafting, the stem of a plant is cut and then fitted on another strong plant and covered with grafting wax. Example, apples, oranges, watermelon, ornamental plants. Tissue culture. It is a collection of techniques used to maintain or grow the plant cells, tissues, or organs under sterile conditions on a nutrient culture medium of known composition. The picture shown here shows the process of grafting. It is basically done for getting different colors of rose flowers in a single rose plant. The diagram here shows the process of layering. Strawberry, lemon, and jasmine are propagated by this method. This picture shows the process of stem cutting. Tissue culture. Tissue culture is a technique of raising new plants from a small portion of a plant tissue over a culture or synthetic medium under sterilized condition. Its various steps are: small piece of plant tissue is taken from the growing tips of plant, such as a tip of root or shoot. The tissue is then transferred to a test tube containing suitable nutrient medium under sterile conditions. This medium contains the plant hormones. Due to the presence of plant hormones, the tissue divides and forms an undifferentiated mass of cells called callus. The callus then differentiates into plantlets. These plantlets are further transferred into pots or soil to grow and attain maturity. Now, the advantages of tissue culture. Number one, it allows rapid multiplication and a large number of plants can be developed in a short period of time. Number two, little space is needed for growing tissue culture. Number three, propagation of tissue culture can be carried out throughout the year, irrespective of seasons. Number four, the technique of tissue culture is used for production of ornamental plants like orchids, dahlia, carnations, etc. Now, these are the assignment questions to be done in the notebooks. Thank you.